In this interaction, we'll discuss about basics of complexation method of estimation, its mathematical expression, and solubility change method for estimation of complexation. Now, if you observe the equation mentioned over this slide, so capital M, which is representing the uncomplex metal ion, A is the donor or ligand used, and Ma base to the N, which is representing the formed complex, where N is the number of moles of ligand which binds to 1 mole of metal ion. So if we write stability constant for the above mentioned equation, it will be in numerator the product, which is the complex form between metal ion and ligand mentioned in the numerator, whereas uncomplex metal ion and uncomplex donor or ligand mentioned in the denominator. So in this manner, complex stability constant can be expressed. Next, if we transform this onto the log scale, so the equation will be log complex, which is Mn, is equal to log K plus log metal ion concentration plus N log concentration of ligand or donor ions. Now, if you observe in this equation, the highlighted one, which we generally estimate in complexation. So these two factors which were highlighted in the equation are stoichiometric ratio, which is the ligand to metal or donor to acceptor ratio. Then stability constant of complex, which is the K value. So mainly in estimation of complexes, we determine the stability constant value and the N value, stoichiometric ratio value, which is ratio of ligand to metal or donor to acceptor. Now coming to the solubility method for estimation of complexation. Now consider the example of para-aminobenzoic acid with caffeine. So para-aminobenzoic acid, if we complex it with caffeine, in that case generally the solubility of para-aminobenzoic acid increases due to complexation with caffeine. We consider this example for estimation of complexation. Now, initially in this method, we take different concentrations of caffeine in series of flasks, followed by we add excess amount of para-aminobenzoic acid in each flask. We agitate it. After a certain period of time, we filter it and we analyze the content of para-aminobenzoic acid for each flask. Now, by getting those datas of or those concentrations of para-aminobenzoic acid, we plot graph of as observed in the given diagram on slide. On y-axis, molar concentration of para-aminobenzoic acid, which we determine, versus molar concentration of caffeine. Now, if we observe this plot at point A, which indicates solubility of para-aminobenzoic acid in water in absence of caffeine or at zero concentration of caffeine, means whatever the original solubility of para-aminobenzoic acid, that data initially we plot. Then further, if we observe from point A to point B, the concentration of para-aminobenzoic acid is increasing with increasing amount of caffeine, representing that there is increase in solubility with complexation and the amount is increasing from point A to point B. Solubility which is increasing. Now from point B to C, if you observe, there is saturation, no change in concentration of para-aminobenzoic acid even with increasing the concentration of caffeine, which is also represented as saturation point. Beyond C, if we proceed, then there is decrease in the concentration of para-aminobenzoic acid, because due to formation of higher complexes, all excess solid acid get converted to complex, and because of the formation of higher complexes, there is decrease in the concentration or solubility. Then if we calculate the stoichiometric ratio and stability constant, it will be calculated as we all know that the stoichiometric ratio, which we have seen in previous slide, is the ratio of donor versus acceptor. So in the case of para-aminobenzoic acid caffeine complex, we represent it as, in numerator, concentration of caffeine in complex, whereas in denominator, concentration of PABA in complex. In this manner, stoichiometric ratio we can represent, which is ratio of donor to acceptor. So here in this example of caffeine PABA, caffeine is acting as donor, whereas para-aminobenzoic acid is acting as acceptor. Now how we can calculate these two terms of numerator and denominator? So concentration of caffeine in plateau region can be calculated by simply subtracting concentration of caffeine at point C, which can be obtained from x-axis, and concentration of caffeine at point B. If we subtract these two values, we can get concentration of caffeine in plateau region. Secondly, PABA in complex, how we can calculate it? So simply total para-aminobenzoic acid which we have taken initially, subtracted by concentration of para-aminobenzoic acid at point B or C. Simply by interpolating it on y-axis, we can get this concentration of para-aminobenzoic acid. So total amount of PABA subtracted by PABA of this point of either B or C, this value, if we subtract, we can get concentration of para-aminobenzoic acid in complex, and by taking these two values, we can be able to calculate the stoichiometric ratio. After stoichiometric ratio, for calculation of stability constant, now stability constant expression also we have seen in the previous slide, which we can represent in the form of product in numerator and reactant in denominator. So our product is the complex, that is PABA-caffeine complex, versus uncomplex para-aminobenzoic acid and uncomplex caffeine. In this manner, we can calculate the stability constant. So, PABA-caffeine complex, how will calculate? Simply, para-aminobenzoic acid at point B, this value subtracted by para-aminobenzoic acid at A point, this value. Means simply by taking difference of this, we can calculate that during complexation how much para-aminobenzoic acid enters into the complex or form complex with caffeine simply by subtracting para-aminobenzoic acid amount at point B with para-aminobenzoic acid at point A. Then uncomplex para-aminobenzoic acid, simply we can take the value of para-aminobenzoic acid of point A, which represents uncomplex para-aminobenzoic acid. Simply PABA value at A point, which represents uncomplex para-aminobenzoic acid. Similarly, caffeine uncomplex we can calculate by taking into consideration the total caffeine which we have taken subtracted by caffeine entered into the complex. Now caffeine entered into the complex, for that we can consider the value of PABA caffeine. That PABA caffeine complex indicates the caffeine entered into the complex or formed complex with para-aminobenzoic acid. So simply total caffeine subtracted by caffeine which formed complex with para-aminobenzoic acid, by taking the difference of these two values, we can calculate uncomplexed caffeine. And simply by calculating the uncomplexed para-aminobenzoic acid and uncomplexed caffeine, we can, by putting these values into the equation, we can be able to calculate the stability constant. So in this manner, by change in solubility or by increase in solubility, we can calculate the stoichiometric ratio as well as stability constant for the mentioned example of para-aminobenzoic acid and caffeine, and can estimate the complexation.